Good morning and warm welcome to all the participants. Today we have the pleasure of having Dr. Manish Kumar with us. Sir is Associate Professor and Head, Department of Biophysics, University of Delhi, South Campus. And the topic chosen by our esteemed guest is evolution of antibiotic resistance, how it spreads. Sir, you are audible as well as visible, and your presentation is also visible. You may start, sir.
Okay, good morning, everyone. And first, I would like to thank the organizers for inviting me to share my work in this program. So, I guess this is a refresher program, and all the participants are teachers. Am I correct, Dr. Sir?
Yes, sir. Most of the story.
Sir,
Yes, sir.
yes, sir.
Most.
Participants are
Hello,
joining
can
in,
you hear
and
me?
they will. Yeah, yeah, we can
Yes,
hear.
sir.
My voice is clear?
Yes,
Yes,
yes.
sir.
Okay, okay. So basically, today I am going to discuss about uh, my work, which is related to the evolution and the spread of antibiotic resistance. So, first, let me discuss what is the antibiotic resistance. Although I believe that this is not something which is new in a field of agriculture, usage of antibiotics is very widespread, and everybody uses that. Okay, so be in the case of animal husbandry or agriculture. So, if we go into the history of antibiotic resistance, so Alexander Fleming got the Nobel Prize in 1945, and in the time at when he was speaking about his felicitation lecture, he told that the time may come when penicillin can be bought by anyone in the shops. Then there is a danger that the ignorant man may easily underdose himself and by exposing his microbes to non-lethal quantities of the drug that make them resistant. So this was a very beautiful, you can say, prophecy. So this in this chart I have shown in the left hand side, you can see the timeline of discovery of different antibiotics, and the the right panel shows uh, when
Enter your PIN to unlock the device.
when the resistance was first reported. So if you see for the penicillin, it was discovered in 1941, and the first resistant strain against penicillin was discovered in 1942. So similarly to the timeline, you can see it's not just a gap of 1 or 2 years after when the antibiotics were marketed, and then subsequently the resistance strain was discovered. Now coming to the what is the antibiotics resistance. So by definition, it is the relative or complete lack of effect of antibiotics against a previously susceptible microbe. So here you can see this is the CFU in bacteria, and this is the number of days. So you can see here, so when the antibiotic is applied to the culture media that contains the bacteria, so it affects the growth of bacteria and the number reduces drastically. But there over time, but then a point comes after which the antibiotics is not effective at all, and at that point the bacteria again start growing. So basically, this why this has happened, because application of antibiotics has killed all the sensitive bacteria, and the only bacteria that is remaining at this point is those who are resistant to the antibiotics. And then, and now there are many mechanisms and if of antimicrobial resistance. So I have listed here the few common ones. So this is a cartoon that shows antibiotics in form of bullet, and this is the target. So here there is one mechanism that is called as altered target site. So here bacteria antibiotics go inside the bacterial cell and bind and thereby that adopt this mechanism, they basically reduce the penetration of the antibiotics inside the cell. As a effect of that, the antibiotic does not go inside the bacteria and it does not harm them. Then the efflux mechanism is there. So in this, the antibiotics is able to go inside the cell, but pathogen develop a mechanism to efflux out the antibiotics, thereby they become resistant. Then there is a mechanism that which in enzymatic inactivation or modification. So what pathogen does, it enzymatically modify or degrade the antibiotic. So here you can see this is the antibiotics, it goes and carries. So bacteria develop enzymes that it might degrade the antibiotics at all or modify it. And then there are bypass pathway in which bacteria bypass the pathway on the antibiotics work. So just 10 years back, the how much antibiotic resistance is going to affect the human kind, nobody has bothered or they did not want. There was no such study. But recently, EU has come up with a estimated number of deaths that is going to happen due to AMR in 2050. And it has estimated that only in Europe, there could be 10 million deaths due to AMR. So that is more than the death that is caused by the cancer or any other infectious disease. So this is related only to the Europe. This is not related to the. This is not the global scenario or scenario of India. And you know the population of Europe is not as much as in other continents. So we can easily extrapolate this to the number of deaths that might happen due to the spread of AMR. So we need to have be very cautious about this issue. So basically, we have worked in this antibiotic resistance in three different broad areas. So we have developed machine learning and in silico tools for detection, discovery and annotation of beta-lactam family. Then we have developed some tools for the prediction of bacterial efflux protein that can also efflux out the antibiotics from the cell. And then we have developed some machine learning tools for the ARG surveillance for determination, for detection and characterization of antibiotic resistance in different omics data sets. And one thing I want to make it clear here that all these tools are open access tool. Anybody can use it without paying any fee. So what are beta-lactamases? Beta-lactamases are among the most important and widely used antibiotics. However, enzymatic hydrolysis by bacterial beta-lactamases is increasing with day, and that is basically compromising the efficiency of this drug. So when antibiotics was discovered in the late 40s, it was speculated that humankind has won the battle against the infectious disease, but in recent 10-15 years, it has been found that no, the battle is going to start again, because most of the pathogens that were earlier we can treat it easily by the antibiotics, now they are going to be resistant. The other side story is the uh, is a opportunistic pathogen or non-pathogenic microbes that acquire the antibiotic resistant gene and keep it with them. and they can also transfer it to the pathogenic bacteria. So this basically a whole lot of things is going on inside our in all the microbiome or biotic reservoirs where these things are happening. Different pathogens are sharing their genetic material and spreading the antibiotics resistance. So since beta lactams antibiotics are very important in the Indian context, so I started working on beta lactamases. So there are two different classification system of beta lactamases. So it is either the the one class is serine beta lactamases and the second class is metallo beta lactamases. So serine classes are further divided into three other classes A, C, and D, and metallo beta lactamases are class B metallo. So this is basically the Ambler classification. The other more so Ambler classification system is based on the sequence homology. The other popular classification system is on the basis of the function. So it divides the beta lactamases into four groups. So class 2 group is the most prevalent and widespread. So it includes all different type of beta lactamases. So mostly I have worked on I have used Ambler classification system for my work because it is easy to follow due to sequence homology, and we work either on the sequences or the structures. So it gives us a very easy backbone to analyze our result. But I am not saying that this functional classification is not good for us. So different tools for this functional classification system. But whatever I am presenting here is based on Ambler classification system. So when I started this work, I started with development of a database which is called a CVMR. This database from this URL, and CVMR is among the 22 databases that is used by the European Commission for health and consumer safety survey on their market. So this is how the database is built. So the different data fields that it contains its ID, organism name, gene, protein name, so all this information. So when you go to the CVMR, you can browse the data either using the sequence search, or you can do the blast search, or you can browse the whole data set, or you can also use the fingerprint search. So what is fingerprint? I will explain it later on. And then there are with all the beta lactamases family that is involved, that is there in the CVMR. With that, you can get the information about the active site residue, hydrolytic profile, mutational frequency versus variability, fingerprint. So all this information you can get. So when you search CVMR, you will, as a result of such page. So here you can see, this is the protein ID, this is the Ambler class, this is the name of the protein, this is the fingerprint. So fingerprint is basically a small region of amino acid that is present in a specific family of beta lactamases only, and we have used that to recognize the family of beta lactamases. So what are the active sites? So different information you can get from the CVMR. So this is the overall information you can get from that. So you can click over any of this family name and you can get the detailed information of the family. So here this is the variability plot which shows that what are the region that is variable. So it is very useful in that design. So you can target the regions which are variable, and then the phylogenetic tree is also there. So these type of, we have also listed a few key references which we have used during the time of annotation. So this is the blast search. of the CBMR. So you can submit your sequence and you can find out what are the similar type of sequences uh, that are present in our databases. So by doing that, you can annotate your uh, protein. So if you suspect that your protein is a beta lactamase and you want to annotate it, you can use our web server. So this is a comparison between CVMR and other. Yeah. Hello. Is there any question?
No, sir, just some distraction. Please continue.
Oh, oh. So in so this is the CBMR. These are other beta lactamase databases like ADB, ARDB, like Clinic Institute Past. So you can see that there are few things which are uh, present in our database, but that is not uh, present in other database, particularly the conservation profile and the fingerprint, which are absent from all other databases. Also, the number of families are more in our databases. So now coming to the family-specific fingerprints in beta-lactamase family. So basically what we did, we analyzed the conservation pattern of different beta-lactamase families and try to figure out what is there any conserved stage of amino acid which is specific to a particular beta-lactamase family. So for this we have used a technique which is called as expectation maximization approach, and we discovered in few almost 100 families of beta lactamases, we were able to find there is a conserved pattern of amino acid that can be used to uh, recognize that family. So we have used that conserved pattern to recognize the family of the beta lactamases. So you can get the detailed information about this work on uh, mm, this paper. So by the way, this was my first paper as an independent PI. So this, so this is how it looks like. So if you go to the BEL beta lactamase family, there were total 3 sequences, and this was the this figure got this. So this was the fingerprint that we have found. So in the Pro family, there were total 15 sequences on which we have worked, and this was the fingerprint that we have found. So there are total almost 100 we have found fingerprint in almost 100 families of beta lactamases. So this fingerprint we have also used for predicting and annotating the class and family of protein, beta lactamase specifically. So what we did, we have developed a web server in which you can submit your beta lactamase protein, and it basically search whether your protein contains that conjured stage of amino acid or not. And if that stage is present, so the family which is associated with that stage that was assigned. So this is how you get the result. So this is the protein that a user has submitted, and in this particular protein, these fingerprints may be present. So this is the associated e-value, and this is the probable family. So you might ask that why there are 3 families instead of 1. So every protein should belong to only one protein family. Actually, what happens, beta lactamase is a very diverse family. So it never happens that you can get a hit with only a specific uh, beta lactamase family. So even there are many sequences in which these type of sequences are present, but they are slightly different. And one more thing I want to emphasize here is that the searching is not for the exact amino acid. So within this family, there are also conservation and the diversity. So we have to take care of this thing. So if you want a very specific family, so I will recommend that you use the family which has got the least E value. So by that you can bring specific. Yeah. So besides the LACT FP, we have also developed a sequence full protein sequence based prediction method, which is actually a machine learning based method. So we have discussed that there are four classes in in the Ambler classification system of beta lactamases. So this particular system, which we named as bread lactamase, this classify a beta lactam into four different classes. So when we started this work, we have found that there are the only way to annotate or classify the beta lactamases was using the similarity search tools like BLAST or FASTA. So what we used to do, whenever we isolated a new gene of beta lactamases, we submit it into the NCBI BLAST search or FASTA search, and then try to figure out which sequence is the most common, which are matching to the query sequence. So, but there are also many gray areas. So if the conservation is good, there is no problem. But then there are a situation where you could not find a single hit, or there are multiple hits with the same type of e-value. So in that case, basically the user get stuck at to which family he assigns the uh, lactamase. So for this, we have developed a machine learning based tool which take the sequence and classify into the probable Ambler class to which it might belong. So basically, the algorithm works in two different levels. You have the query sequence. So it reformat the sequence into pseudo amino acid composition and submit it to the level 1 classification system, which actually determine whether the input protein is a beta lactamase or not. If it is a beta lactamase, then it goes further down to the level 2. If it is a non-beta lactamase, then program stops here. And if a protein qualifies to be a beta lactamase, then it is divided into the four classes at level 2. So we have also benchmarked the accuracy of this method on a data set, which we called as independent data set that was not used for training the tool. So you can see here that almost in all the protein, all the class of beta lactamases, it has achieved the accuracy of almost 75%. So after some time, we further improved the algorithm of the lact, uh, spread lactamases and make it to classify up to the subfamily level. So in the class B beta lactamases, there are three subfamilies B1, B2, and B3. So the new modified system, which we named as BLA PRED, it takes a sequence, predict whether it is a beta lactamase or not. And if it is a beta lactamase, then at the second level, it predicts whether it belongs to class B. And if it belongs to class B, then further divide into one of the three uh, B subclasses. So whatever I have shared up to you is related to only one or two classes. So this BLA PRED was can able to classify a input beta lactamase into one of the four classes and then one of the three sub classes of the class B. But we know that beta lactamase is a highly diverse enzyme, and within each class there are multiple families, subfamilies. So then we try to find out, is there any way that we can, if we have a sequence from the beta lactamase, then we can classify it into the different families, not only in the different subfamilies, families. So our earlier method can work only up to the class level. So then we try to figure to construct a model that can work even at the family level. So what we did for this, we took combined all the beta lactamases data set from the known data set and divided into different subfamilies. So here also the basic architecture that we used for the classification was Ambler system. So then we divided all the proteins into class A, class B, class C, and D, and then we uh, further divided each of the four classes into different families and subfamilies. So then we build a model for the classification. So this is again a machine learning based model, and then we evaluated the performance on using leave one out cross validation. So leave one out cross validation is basically a method in which we during the time of training we hold one sequence from the training data set and whatever, and then we train the model and evaluate the accuracy of the trained model on the data point which we have withheld from the training. So in this particular, we were able to able to train a machine learning based method that can predict that can take a known beta lactamase protein and it can classify into 106 families of the four classes. So here so there is a web server for that. So in that you can just submit your sequence, select some parameter, and it will classify a input sequence into one of the family of beta lactamase. So whatever I have shared with you till now is development of different tools. So beside developing the tools, we have also used those tools to do the survey of antibiotic resistant genes prevalence in different reservoirs, so be it water bodies or microbiota, all this. So there is a class of pathogen which is called as escape pathogen. So escape pathogens are basically it is a group of pathogens that consist of Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter. So these pathogens are a very leading cause of nosocomial infection, and sometimes and most of these pathogens are highly antibiotic resistant. So then we try to figure out and Obja class of beta lactamase, Obja family of beta lactamase is one of the leading cause of antibiotic resistance in these pathogen. So we try to use our developed tool to analyze that what are the prevalence of different type of Obja variants in the escape pathogen. So what we did for that, we took the different genomes which are there in the NCBI genome databases, then classify the genome into only chromosome. So those genomes, those pathogens which has only chromosome, and those pathogens which contain both chromosome and plasmid as a genomic material. And then we translated the genetic material into the proteins. And then we found that in the in the eFCM, the total number of genome we found in NCBI was approximately 2,200, and the total number of complete genome of beta lactamase is 187. So there were the number of genomes which are present in NCBI and the total number of complete genome was there too much variation in that. So there are almost a majority of genomes which are submitted, which are available in the NCBI genome databases, they are fragmented. Means you can't get a single genomic sequence. You will always, in most of the genome which are present in the NCBI genome databases, they are fragmented. There are many regions which are missing. So then in the complete genomes, we took only the complete genomes and try to find out how many of OBJA beta lactamases are present in them. So as you can see that in the EPCM, the total number of genome, complete genomes were 187, but there was no OBJA variant. In the SOES also, there was no OBJA variant. In the 732 complete genomes of Klebsiella pneumoniae, there were 263 OBJA genes were present. In Acinetobacter, out of the 233 complete genome, 408 beta OBJA variants are present. So in summary, you can say that we got total 2,255 complete genomes of um, escape pathogens, in which 929 variants of OBJA were present. And this also shows, this column shows that the number of proteins which are present in only chromosomal. So you can see here that in the Acinetobacter baumannii, in the chromosomal, in the chromosome, there are 50 different type of genome pair present which contain only the chromosome, and in this chromosome, 82 different beta lactamase, OBJA lactamases are present. But in when you see the genomes which contain both chromosome and plasmid as a genetic material, you can find that most of the genetic material which are present. So this is the distribution. So out of the 69 chromosome, there were 257 beta OBJA lactamases were present in the Klebsiella pneumoniae. In the 183 genomes which contains both chromosome and plasmid as genetic material, 326 beta lactamase, OBJA lactamase was present. So we did all this type of analysis, and then we tried to find out that what is the diversity among all these OBJA lactamases. So for this, we did a evolutionary study, and we try to find out that is there any all the OBJA which are present in escape pathogen, whether they are related with each other or they originated from a what is their evolutionary pattern. So we find on the basis of their location in the genome. So what we found that in the Acinetobacter baumannii, so they are clustered in a single group. In the OBJA which are present in the P. aeruginosa, if they are clustered together, and then there are a region which are present in all the different four species in which OBJA were present. So this shows that there is no clear cut restriction, means it is not happening that a particular OBJA is originated in a particular organism and it is remain confined there. They are interchangeably. So there are two possibilities, that either they are exchanging the between different escape pathogen, or the same type of evolutionary mechanism is working on all the species of escape pathogens. So out of the total this number two, 29 object that we found. So then we also try to figure out. So these OBJA enzymes can be grouped into 16 different groups of OBJA beta lactamases, and in the OBJA, there is a lot of variability between the sequence, between the structure and their hydrolytic profile. So then we try to find out that is there, what are the different in how they are clustered with each other, and how much similarity they are showing are there in the escape pathogens. So this shows that these So our results showed that there are 16 different families of Ogja beta-lactamases which are present in the escape pathogen, and they can be easily uh, discerned from each other on different branches of the phylogenetic tree. So we also try to figure out is there any conjugate pattern among different 16 subfamilies of Ogja which we found in the escape pathogen. So what we found that there are certain regions which are conserved among all the Ogja, and most of these regions are basically they are either the part of catalytic site or important in the um, structure of the enzyme. But this also shows that whatever variability we are seeing in the Ogja, it's it is highly variable enzyme, and it is due to this mutation in the non-essential part, or you can say a not so important part of the protein, is leading to the diversification of Ogja. Then we also studied the time stamp. We also did the time stamping of evolution of different Ogja, and then we find that there are different. So here you can see this is the- from the ancestor, from the common ancestor, the 16 family that we have considered during this study is divided into two different groups, and then again one group is isolated, it's and then the other group is rapidly diversifying. Then we also calculated the pairwise amino acid identity between the 16 subfamilies of Ogja that we found in the escape pathogen, and here so greener is the um, color, more is the similarity. So here you can find, so this is the diagonal, it means the similarity between the same subfamily of Ogja. So here you can see that there are a lot of variability among different families of Ogja that we found in the escape pathogen. So they are basically isolated groups, and this also indicates, this along with this tree indicates that they are evolving in a divergent way. So we also calculated the root mean square deviation, which is popularly known as RMSD deviation between the different uh, between the structures of the subfamilies. So here again you can see that there is lot of variability in the structure also. So now coming to the prediction and classification of, so whatever I have discussed, that is basically related to the beta-lactamase. Uh, so we have also, after doing this work, we have also started working on the efflux antibiotic resistance genes, which basically use efflux property of the protein to classify, to impart the antibiotic resistance. So for that, we have developed a machine learning tool. So basically in that tool, what we have done, we have taken all the efflux proteins which are present in the prokaryotic genome and try to figure out which proteins can probably also efflux out the antibiotics. So we have taken 4 different families of efflux protein and then developed machine learning model to figure out which are among them and able to predict the to efflux out the antibiotic resistance. So this is the web server. So you can access this web server from this URL, and if you want to read the paper in detail, you can go to this particular paper. A copy is also you can find in this web page. So basically, if you have isolated a efflux protein and you are trying to figure out whether there is any antibiotic resistant capability in your efflux protein, you can use this web server. So now coming to the so basically both beta lactamases and efflux protein, they are isolated category of protein, and we know that antibiotic resistance phenomena is not a isolated phenomena. So almost all the biotic and abiotic factors are involved in that. So the the popular myth is that it is only the medicinal use of antibiotics that is bringing the antibiotic resistance, but in actually it is not. So it is actually the use in the livestock industry and the agriculture sector which is bringing up the antibiotic resistance. And as a antibiotic resistant gene is evolved anywhere in the food chain, it ultimately spreads to all the factors which are involved in the food chain. So to analyze this behavior, we try to develop a machine learning method so that we can take any high throughput data and can figure out what is the variability of the antibiotic resistance in that sample. So I'm going to discuss about a tool that we have developed to uh, characterize the different antibiotic resistance genes in any high throughput data, be it the genomic data or proteomic data. So the name of the tool is BACAR scan. So to develop this tool, what we did, we collected all the antibiotic resistant genes which are known so far from different databases, and we used clustering techniques to develop the cluster. So on the basis of different cluster that we found, we developed the hidden Markov model, which is basically a probabilistic model to model the evolution of a closed group of sequences. And then ultimately we find total there are 254 HMM models that we so basically these 254 HMM models encompasses all the known diversity of the antibiotic resistance gene. So the overall architecture of this tool is that if you have a new sequence, so you have to select whether the sequence is a gene or protein. So if it is a gene, then you can either choose it to translate into the protein and use the model which were developed on the protein, or you can use the gene model also. So for each input sequence, you will get a notation. So what is the probable family of the gene? What is its probable function? In what type of hydrolytic or resistance profile it has? So all the information you can get. So we have benchmarked our method on different popular uh, classification system, for example, ARDV, response, and backyard. So this is a benchmarking study that we did in human gut microbiome. So here you can see this is the comparative evolution of these three ARDV, respan, and backyard scan. So this is the backyard performance of backyard scan. So in most of the classification, most of the families, our method has worked almost equal or better than the other popular methods. So we have also there is a method which is a database which is called as CART, which is a very popular database. So we have also benchmarked our method in comparison to CART, and here you can see this is the red bar denote backyard scan and blue bar denote CART. So here you can see that our method is almost as good as the CART, or in some cases it is better than that. So we have also annotated the proteome of escape five sample strains of escape pathogen. So here you can see. So the overall trend of different type of antibiotic resistance gene is almost similar in all the by all the tools. So this different colors show the different type of antibiotic resistance genes. So we have also benchmarked our method on a clinical metagenomic data. So here also, so this different colors denote different type of antibiotic resistance mechanism, and this is the number of count of gene in each of the patients. So there were total 6 patient sample, and we have annotated the different uh, type of antibiotic resistant gene in each sample. So there is a different database which is called as RESPAM that also use the same strategy that we have used for developing a KRS scan. So we benchmarked our method on basis of that, and this yellow golden bar indicates the BACARS scan, blue bar indicates the RESPAM. So here you can see that in some cases our method is working better, in some cases RESPAM is working better. So means this shows that our method is as good or as bad as other method. So there is no clear-cut winner, but there also our method is not inferior than others. So this is the uh, distribution of different type of uh, antibiotic resistance class that is there in the our database. And this is how our method is performed when we use leave one out cross validation on the data set. So this is the web page of our method. So you can submit any. So right now we have there is no provision to submit the whole proteome or whole genome. So for that you have to download the standalone version. But if you have a 10, 20, 50 genes, then you can genes or protein you can do at our web server. So this is the end of my presentation. So I would like to thank my collaborator, Professor J.S. Vidhi, Dr. Manisha Goyal, and Dr. Nita Singer, who are part of this research activity, and the team of students who contributed in that, and Dr. Suhani Jain, Dr. Ravindra Kumar, Dr. Vandana. So Suhani Jain was my initial MPhil student, so she is a research program officer in Australia. Dr. Ravinder, he is a scientist in right now in NIH. Dr. Vandana, so she has moved to cancer now, and she is working as a scientist in Institute of Human Genome, so, which is part of CNRI in France. Dr. Avisika, she has moved to McGill University, and there she is doing the metagenomic work and characterization of different type of antibiotics genes in different metagenomes. So she is actually who worked on antibiotic resistance. Others have basically aided her. And Anjali Diksha, so Anjali had just submitted at Viva. So she is now moving to University of Washington next week. Diksha, so she has also carried forward the work of Avisika, and so she is also going to submit her. This is soon. The other student who has contributed, Manisha Aswal and Govind Kaburu. So the funding for this work is mostly come from ICMR, UGC, Delhi University, and some part was also given by BIRAC and Innovate UK. So thank you. If there is any question, I would be happy to answer that.
Thank you, sir. Now, if we have any questions, any queries, or any interaction you would want to do, please turn on your microphones or type the query or interaction in the chat so that we can reply or revert to it. Dr. Jaya Kumari Annamalai has raised her hand. Do you want to say something? Dr. Jaya Kumari,
No, sir. No, sir.
you want to say something? You want to ask something? Dr. Jaya Kumari, you want to say
Yes,
something?
sir. No, sir. I think mistake. No, sir. It's okay, sir. Clear.
Anybody else who wants to interact or ask a question or has a query regarding the topic of discussion?
So I would have no problem if you email me the question, or if you want any specific type of information, or if you have any sample that in which you want to characterize the prevalence of antibiotic resistance genes. So I would be happy to help you.
Any interaction, any question we have, then we can discuss. We have few minutes before our time finishes up. Sir, I don't think there are any questions from the participants. On behalf of the organizing committee as well as on behalf of the participants, we extend our gratitude, sir, for being with us with this lovely presentation on an apt topic which we are dealing with on a daily basis. And we feel like at times people do not get well soon. Why? Because they have been using a higher, in humans, antibiotic resistance is found. So, on behalf of the organizing committee and again on behalf of the participants, I would like to extend our heartfelt gratitude, sir. For thank you for being with us. Thank you, sir.
Thank you, Dr. Asar, for inviting me to share my work.
Thank you, everyone. Let's join back at 2:30 for the next session.